as important to the workings of the system and not just something to be removed. I mean, it seems like a very simple idea, but in many cases, we always want subjects to behave the same way and to give the same signal every time. And it really helps if we can reduce variability. And if subjects are going to vary from trial to trial and moment to moment, it does cause all sorts of issues with our statistics. So trying to make use of that as opposed to trying to remove it, I think is really an important point to keep in mind.
Right, absolutely. And I think too, and I sort of, we'll get to this in a minute, but I think that also depending on what the brain is sort of being tasked with doing in a given moment. So if it's, faithfully representing a sensory signal, well, that it's important for that to be stable and consistent each time a participant is responding to that, as opposed to a task that sort of requires some flexibility and some metacognition, for example, where perhaps variability is beneficial to the system as opposed to detrimental. So I think it's also just kind of interesting to think about instances in which it actually would be helpful as opposed to harmful. And thank you, Armin, for reminding me to record. I still forgot. So sorry about that, the first couple of minutes, but we are recording now. All right, so just to go into a couple studies, the first of which is that this is where, sorry about that. This is really where a lot of this variability work came from, is that there's a long line of studies really looking in terms of cognitive aging and thinking about variability across the lifespan and how it changes. This is a paper from Lucina Udin's group and other colleagues looking at across the lifespan how variability is actually changing across adulthood. And it tends to be that variability goes up in some networks, but it actually goes down in others. And this is sort of, in cognitive aging, there tends to be more of a focus on the decrease in variability over time within different networks. I chose this figure because I think it's nicely illustrative of how it really depends on the network that you're looking at. Some can go up and some can go down. But that in terms of behavior, that perhaps there's this sort of optimal range, sort of having some variability in the system for optimal behavior. But if there's too much or too little, then things can be problematic. And so a lot of the research tends to focus on how variability is changing over time within different networks, and then also how that relates to behavior. And I chose this particular study in terms of links to behavior because I think it's a really nice finding where actually what the authors found here is within a brain region, in this case, the inferior frontal junction, depending on the task, it was the same set of participants in these two figures. Here, variability had a negative relationship with behavior, whereas here it had a positive one. So that it can actually flip. So the direction between variability and behavior can flip, even within the same brain region and the same set of participants, depending on the task they're being asked to do. So Jess, this kind of goes back to what we were talking about a few minutes ago, that sort of what the task is can dictate a lot what this relationship looks like. And then to further build on that, this is a study that I had done a few years ago with some collaborators, in which here we looked at individual differences. So we looked at reading skill out of the scanner, measured using different reading tasks in children, and then related it to variability in an fMRI task while children were reading words. And here we found a positive relationship between variability and reading, which was actually in the left inferior frontal gyrus. Directly opposed to an earlier finding using subcortical auditory brainstem responses, in which the authors had found that variability was, or that the response consistency, meaning that variability was actually lowest or most consistent in the children who had the highest reading levels. So they actually found a reverse relationship between variability and reading from what we found. And again, I think this illustrates that there's some connection to individual differences, but it's also important to think about what might be driving that. So here, this was a low-level responses to speech in which the participants were not asked to do anything, as opposed to a high-level reading task. And again, this was found in subcortical areas, whereas we found a positive relationship in the IFG, which is a fairly higher level area. So, I think it's just helpful to think about how variability can be interpreted within these different experiments. So yeah, Armin's question, how do we calculate variability? So here we did the standard deviation of the bold response across the fMRI time series. We actually got beta values from a GLM. So this was an event-related design. So we had single trial beta values, and then we took the standard deviation across those. Here in the Hornicle and Krauss study, the authors actually took like the odd trials and even trials and basically calculated the correlation between the two to see how consistent the responses were. So I'm hoping that kind of gives you a taste of sort of some of the results that one can get here, how they can sometimes be inconsistent, and perhaps some of the reasons why, and some of the things we need to be thinking about. And so I think that leads nicely into Stephanie's presentation as another example of what this variability kind of information can tell us and what we can ask with this kind of design. So Stephanie, I'll turn it over to you if you could get your presentation set up.
Sure. Okay. Okay, so hopefully everyone can see my screen. Okay, great. So, like Dr. Malins mentioned, my name is Stephanie. I'm a second year PhD student in the clinical neuropsychology program at Georgia State. And I'm working with Dr. Malins on my master's thesis, looking at within individual neural variability and specifically in the MBAC task and behavioral performance on neuropsychological assessments, but we have yet to get there. We've been working pretty diligently all summer and the beginning of this semester, and we have some preliminary results. So that is what I'll be talking about today. So the first question that I asked is really, is there a meaningful connection between mean signal measurements and variability measurements? And so the first question is really, what is the relationship between mean signal and within individual neural variability across task runs? So this is looking at across the entire task. My second question is there an overall relationship within individual neural variability and measures of mean reaction time and mean accuracy on the task? And then more than looking at it overall, I was also interested to see if the methodology that we've used would produce any meaningful results regarding specific brain regions. And so the third question that I had is there an association between variability and mean reaction time and mean accuracy in specific regions of interest that have previously been linked to working memory and vigilance, which are two constructs that are measured in the MBAC task. And so for the methods, I'm using a sample of 48 undergraduate students from Georgia State University. And in the scanner, these participants did a letter MBAC task. And so very quickly, the premise of this task is there are different types of trials. So in zero-back trials, the participant is first presented with a screen identifying a target letter. And then they're asked to indicate yes with a button box when the target is subsequently presented, irrespective of the letter case, so whether it's capital or lowercase. In one-back trials, the participant responds yes or no if the letter repeats itself from the stimulus directly before it. For two-back trials, they look to see if the letter is repeated 2 before, and 3-back to look to see if the letter is repeated from 3 before. And so a lot of our work has been done with AFNI and all of the processing that's related to calculate variability. And so we were predominantly using the model given by Garrett and his colleagues for the 2010 and 2011 papers that they did. And so first, we regressed out white matter, cerebrospinal fluid, and 6 motion parameters, which were the X, Y, and Z translations and rotations from the original voxel time series. And then from that general linear model, we got residual values. And so for mean and standard deviation, we were calculating using the residual time series. So for mean, we took the mean signal for that residual time series for each run, and then computed the mean signal across runs for each stimulus type. And then for within individual neural variability, we operationalize this like Garrett and colleagues have done, looking at the standard deviation of the signal and like what Dr. Malins had looked at in his paper. And we concatenated across runs for each stimulus type and calculated the standard deviation. And finally, for each ROI, so I selected 8 total, I created a mask for each of them. And this is a little bit more information about those ROIs. So these have been identified previously in meta-analyses looking at working memory and vigilance in the MBAC task. And so I chose regions in the dorsolateral prefrontal cortex, ventrolateral prefrontal cortex, anterior cingulate, the dorsal inferior parietal cortex, and medial and lateral posterior parietal cortices. And so for results, we ran initially some Pearson correlations to assess first the overall association between mean signal and within individual neural variability for these 8 ROIs across the task. And we found that they were significantly positively correlated. So there's some relationship between mean signal and variability for this task in these participants. Secondly, we also ran correlations looking at variability and task performance defined by reaction time and accuracy. And we found that variability was not significantly correlated with mean reaction time. However, it was significantly negatively correlated with mean accuracy on this task, which suggests that variability in these regions might not be as beneficial for accurate performance, which is different from other results that have said that variability could be associated with greater performance. And then finally, we ran correlations for each ROI and each stimulus type, looking at the relationship between variability and reaction time and accuracy. And there were no significant correlations in any of these identified ROIs for each stimulus type and with mean reaction time and mean accuracy on the task, which suggests that looking potentially at very specific ROIs in specific stimulus types could be too limited, and variability may be more meaningful if it's looked at more globally for the task. And so some more general implications from these preliminary results. I think that variability methodology is very relevant and a new perspective in looking at this stuff through cognitive neuroscience and also for neuropsychological research and assessment. And actually, a very recent paper came out discussing variability and how it relates to performance on the digit span task, which is a neuropsychological assessment of working memory. So there is an active interest in the field right now, and I'm excited to contribute to that literature. These preliminary results suggest that within individual neural variability might be important to overall accuracy, but potentially not reaction time on the task. And again, this ROI-based approach may be too limited, so the next steps for my thesis are to look at a whole brain cluster-wise analysis to identify broader regions that might be implicated in accuracy and reaction time on this task, more so than specific ROIs. And that is it for now. So if anyone has any questions, I can try my best to answer some.
Great, thank you, Stephanie. I see one on the chat here from Armin. He asked, is variability referring to the bold signal variability across time?
Yes, so stop sharing my screen. So yes, so we looked at the bold variability across the entire task and then took the standard deviation.
Is there a reason you didn't take the bold signal within the relationship to the actual task or the variability related to the actual task as opposed to the variability overall?
So, okay, so actually I answered that incorrectly. So we did identify the TRs that were associated with task and with the stimulus type. So we did get rid of the outside volumes that were not associated with the task.
So you're specifically looking for the variability in response to the task, not the change in variability from between, but the quote unquote rest or baseline condition.
Exactly.
Which is one of the measures that I think either Garrett or maybe Lucina highlighted in their paper, that the ability to switch out of a variable state to a non-variable state correlated to task responses and accuracies.
Mm-hmm.
And just to point out, there are studies showing that during the rest before task can in fact predict the outcome of the task. So
yeah,
it's interesting if you look at both during the rest and during task.
I agree.
And as I point out, I like the hypothesis driven that you chose like a specific region. Although all my work is like a whole brain connectivity, but I really like this type of focus targeted studies. It's very interesting.
Thanks.
Thanks for these great presentations and for letting me jump in on the meeting. I'm still on the email list, so I saw this topic and thought I can't miss it. But so you see a lot of people use the term variability to describe, different aspects of the signal, as have talked about here. So I'm still trying to think about, you have the bold signal variability, which you can calculate during task or rest. Then you have things like trial to trial variability and other, aspects that like Jessica said in the chat here, could be related to that bold signal variability. But I think there's, I don't know if there's any really consistent finding on this, like does bold signal variability enable trial to trial variability, or, does anyone know of that work?
I wouldn't be able to point you to a reference. I could have sworn that I've seen that, but I may be going off of intuition and cognitive bias.
Exactly. I have the intuition that they're related, but I haven't seen any empirical work on it.
Yeah, I think I saw a study, but it was not in activity, in the connectivity from Sepide. I can share the paper here. She was looking at as far as I recall, dynamic connectivity during the rest, and they're trying to see, I might be wrong, but I think she was trying to see how it predicts the task and the variable trial to trial. That's what I recall. I'm not sure. I'm going to share the paper right now.
Great, thank you.
Oh, I remember that work. Yeah, I think it was looking at the pre-trial signal levels influencing the. I don't think it was variability per se. That's why I'm curious.
I think it's like the mean or something.
Yeah,
like if you have a high mean, you can't increase as much and things like that.
Exactly. Yeah.
Yeah.
Right. And one comment I wanted to make too, so it's even in terms of, some of the work that we had done was an event related design. So that reading study I presented we got single trial betas in this event related design. What Stephanie was instead presenting was a block design for the NBAC. And so sort of, making a time series based on the blocks of that particular stimulus type. But that's still a bit different than looking at the standard deviation of single trial bold. And sort of thinking about, because we were sort of struggling with this, is that, indexing spontaneous activity over time? Is that indexing perhaps differences in the actual representation of the words? So if children might have, sort of representations that are more variable, that's perhaps why you see differences in activity of that region. We weren't quite sure exactly what that index is in terms of single trial responses in a task. And I think, I'm not sure that there's a lot of, if there is, I'm not too aware of it either, because I think that's really important to try and see how these map onto each other.
Yeah. Well, I think it's rather challenging that there are so many choices of what you could measure in this case, because we don't have sort of an underlying mechanistic model that says, okay, really, if what you're looking for is trial to trial, changes in neural representation, you need the trial to trial, MSE or something like that. And so you're kind of, there are too many options in terms of what you could measure to look at it.
Right. And I'm not sure if this exists either, but I'd be curious to see if folks have done simultaneous EEG fMRI, for example, to look at variability indices, across the two. So if you look at multi-scale entropy from EEG and then bold standard deviation from fMRI, like how do those map onto each other for the same task, for example?
In the Cobre data set, we had both MEG and fMRI for a lot of the subjects.
Okay.
And I thought somebody had looked at entropy, but maybe not. But we do have data that could answer that in healthy controls or in patients with schizophrenia. So they're not simultaneous, but they are the same tasks and the same subjects over the course of maybe 6 weeks or something like that.
Great. Yeah, I think that would be really interesting to look at.
So for your PhD, Stephanie.
A lot of these we've already talked about, but I just wanted to have them up on the screen because there's so many differences here. I think that can lead to across studies. We haven't talked much about pre-processing, but I think that can have a strong impact. That Stephanie, and as we know we grappling a lot with head motion and what to do with it, and how that can influence some of these signals and how to handle physiological artifacts and so on.
Well, I was particularly intrigued. I think it was Lucina who, in your paper, that said global signal, was thought for a long time to be just noise, take it out, and then there were statistical reasons to, not take it out, and then there's neurophysiological reasons to not take it out.
Yeah, it's kind of a mess, and we're still not sure what to do about it in many ways, because you will for sure remove some important signal if you take it out, but you'll for sure leave the noise in the data if you don't take it out.
Exactly. It's something of a sledgehammer for removing artifacts, but you might remove stuff you want. I'm sorry that Robin Miller isn't on this, because she's of course been doing a lot of the spectral frequency analyses at multiple spectral frequencies, which Garrett noted is sort of an alternative approach to looking at variability. And I would be really interested in her take and whether what she's sort of capitalizing on there is capturing the same thing as the signal variability at different timescales. I don't have an intuition about that.
I like the idea of the EEG fMRI comparison, because you can get around some of these fMRI pre-processing things if you can show some correspondence between the modalities. Because the fMRI, the cleaning we do to do an MSSD study is so aggressive, because it has to be, otherwise people will say, well, you're just looking at noise patterns, and we don't know what that is. So yeah, I would love to see someone do that, the simultaneous EEG fMRI.
Does it have to be simultaneous is my question.
No, maybe it doesn't have to be.
Right? As long as it's on the same subjects and you don't have too much, fatigue or something, you would expect the brain states to be roughly the same. Or if you had enough subjects or you did it, it wouldn't necessarily have to be at the same time. Because that introduces so much artifact in the EEG that I would worry about that. To do it at the same time.
Yeah. I actually, one of my colleagues, Nathan Sprang, is working on something where he's looking at associations between MSSD and functional connectivity, because trying to make the case that they're important, they somehow might influence each other. And he's actually found that across the brain, the regions that have similar MSSD patterns have higher functional connectivity among them. So I think that's another interesting thing, trying to link these different levels of measurements.
I just wanted to jump in. That's really interesting that Dr. Sprang is working on this, because he used to work at Cornell, where I did my undergrad, and he taught the lecture about neuropsychology, and that is what I'm doing now. So
too funny.
Yeah, he actually, he and I and Doug Garrett were at a ski conference in Switzerland a few years ago, back when people still traveled. And, we talked a lot about these variability issues back then. And so we've been working together on a few things, but, we haven't got anything out yet. But my question has been like, if you have this nice measure of variability, how does it relate to all these other measures that people care a lot about, connectivity being one of them?
Mm-hmm.
Because certainly digging into, from my experience with fMRI, I started with spinal cord injuries. And one of the first things that we noticed, because we were doing a lot of hand processing of the fMRI data in those days, was that the folks with spinal cord injury, even though we were getting activity from the same brain regions, just showed much, much higher variability than did the age-matched and gender-matched healthy controls. And then, of course, we see the same thing in schizophrenia. We see the same thing in Alzheimer's disease. So to me, a nice, clean fMRI signal is a sign of brain health, whereas a sign that, if it's bouncing all over the place, it's like, okay, there's something that's not connecting properly. And so trying to force myself to think of it as actually a source of a good thing, as opposed to something you want to minimize, or return to quote unquote health, is really a different kind of approach. And I don't know how to disentangle that, right? Is there something about, there's no question that in various diseases and disorders, we have increased variability if you just look at how the bold signal goes from trial to trial. So what is the mechanism that is driving it to be adaptive, and what is the mechanism that is driving it to be dysfunctional, in a sense?
Yeah, that's a great question, because the same thing happens in autism and ADHD if you look at the literature. And I actually think it's about optimal levels of variability for a particular function. Like the Armbruster papers, I think there were some by the same group that suggest for cognitive flexibility, variability was better for those tasks, but for stability or cognitive control, it was worse, right? So it's like function dependent and brain region dependent.
And that's what you were saying, Jeff, with the picture that you had of sometimes it's good and sometimes it's bad, even within the same population.
Right, exactly. And I think about sort of the sensory cognitive that I have on the screen there too, in terms of the sort of situations under which it's. And so what I've seen in some papers looking at working with individuals with autism spectrum disorder, for example, that it's higher variability for sensory processing.
And certainly with spinal cord injury, that's what I was looking at, with sensory motor processing, largely.
So
with schizophrenia, it's cognitive. It's cognitive as well. And with Alzheimer's disease, it's cognitive as well, not just the sensory.
Right. I just meant in terms of the variability relationship was sort of greater variability in sensory processing tasks specifically. But I
think there's something there.
Yeah, one reason we had thought reading might be an interesting test case is that it involves sort of sensory and cognitive components to it. So I think I'd sort of be curious about what tasks have been looked at as well.
So I just want to understand better what Lucina was saying. So I was wondering if we can talk a little about that. So I think the question was, her comment or question was not clear to me probably. So I was wondering if she doesn't mind to elaborate a little more so I can understand better.
Sure. Yeah, I actually was thinking, there's a lot of literature on trial to trial variability, which means the reaction time or some kind of measure is different from the first trial to the second to the third to the fourth to the fifth. And you see that in ADHD, for example, or a lot of clinical populations where they say, okay, they're just very variable in their responses. But I was thinking that that's maybe slightly different, maybe related, but the bold signal variability that we've been talking about here is just sort of time point to time point, TR by TR variability, which is another thing. It's also variability, but is it somehow related to that behavioral trial to trial thing, which people also call variability?
Okay. So my question right now is, so just to first make sure I understood. So there are changes between different trials, right? And the question is if these are because of. So we also see changes during the scan, even in the rest. And there was also a study that's showing, in fact, they were saying like we gradually change even during the rest. If you chunk a rest to a different trial, it's not trial, but if we chunk in different time point chunk, we see changes over the time. So my question is, if these trial to trial changes is for the same, coming from the same, come from the same source or not? I misunderstood your question, honestly, for the first time when you asked. Thanks for clarifying.
Yeah, exactly. That was my same question: is are these all the same sort of things that we're measuring, or are they just totally different phenomenon? I mean, Jessica had an answer in the chat, and it's true. I think they're probably related, but I don't know.
Don't know. Yeah, don't know.
Is there a way that we can measure the trial changes correlation? I haven't done that. I'm just wondering if they have done like correlate the changes in the trial to trial with the person response, for example, to see if they are correlated or not. I still doesn't answer that question, but I was just wondering if there is a way to, if people have done that.
So I think that Garrett's group has done some of that, and Shale Grady and others as well, where they've looked at standard deviation of the reaction time and how that goes with the standard deviation of single trial hold. Yeah. There's actually an inverse relationship. So the folks that have greater variability in the brain have more consistent reaction times. So that variability is more accurate and more consistent if variability is greater in the brain. So that was sort of one of their major findings, is that there's this counterintuitive kind of relationship there.
Yeah, there was a large discussion about attention in, I think, the Garrett paper as well, and the ability to pay attention. Well, depending on what kind of attention we're talking about. I think they were specifically talking about vigilance as opposed to task switching kinds of attention.
But so that is a counterintuitive, the point you just made about
very much so.
Yeah, wow. That's why I wonder if we're all using variability to mean exactly the same thing or not.
Right.
I'm gonna put my money on not.
As always.
Exactly.
And I think noise is not used the same way either. That's been sort of a quibble of mine. It works in the area of reading, for example, that there's this idea that a noisy brain and sort of, again, it's meant sort of with stimuli coming in and how the fidelity with which they're represented. That's quite a bit different than thinking about variability in the way that we've been discussing it here. But I think those are often used synonymously as well.
Yeah, it definitely helps to be precise there. And I think this repeated question of what are we talking about when we talk about variability? Which variability are we measuring? Yeah, it's very important.
And one thing I wanted to put on the table too is that I think linking this to neurochemistry as well. I think there's a few studies out there looking at, for example, dopamine transmission and or thinking about glutamate and GABA levels, for example. But I think that kind of data would help to try and clarify some of this.
Yeah, I'm not as familiar with the computational work, but I feel like Randy McIntosh's group has done some work in this area, and I think you can do a better job of maybe teasing apart things like noise and variability and whatever when you're just creating the data yourself and then asking sort of some questions. So my questions would be if you have these different brain regions with different levels of variability, how do they interact with each other, or how does that influence the connectivity between them is or some of the questions I would have.
Right. And one thing I'm really curious about too is how this might relate to modularity as well. So it's sort of, again, thinking about learning, and perhaps there's like, if things are encapsulated within a certain network where it can kind of play around with different states and entertaining different possibilities and being more variable, but then you've got other systems which are stable and consistent and sort of the extent to which those are separate from each other. I'm just kind of curious about how that might fit in too.
Yeah, does variability contribute to making these sort of communities or, segregating networks or, like I think there's just so many open questions.
I agree.
And I think that might relate to some of what Stephanie presented with sort of your data looked like it was more of a global kind of thing as opposed to region dependent in that case. And or it could just be, it could be the task, could be the ROIs that were chosen. But I think it's, and in that task, perhaps, like, do you think it's related to what the NBAC task requires, that why variability might not be as beneficial in that case?
Yeah, I think it's an interesting question. And I think, like what we were talking about before in terms of flexibility versus stability too, for something like the MBAC task could be important. And so I think I guess what the MBAC task is measuring, like working memory vigilance and variability, are like kind of new areas that are being looked at. And so I guess we don't really know what the implications of variability are for those cognitive functions. And so I think it's exciting to start looking at that. Like I said, there's one paper from Boylan and colleagues that was just published looking at working memory and variability, and they had some different results than what I figured out. They used a digit MBAC span MBAC task, and then also looked at digit span. So I think it could be a factor of the population that we're using, a factor of maybe it's the letter MBAC task that is different in some way than a digit task. But yeah, that's a pretty recent paper, so I definitely need to look more at it.
Yeah.
So Jessica, sorry, go ahead.
I was going to say, it's illustrative of how much interest there is in this area.
Very much so.
So I just saw a comment from Jessica. I haven't. So are you saying if there is a way that we can see dynamic variability predict those type of task score, correct?
What I was wondering about is if when we're doing dynamic functional network connectivity, we're usually looking we're looking in different ways at how the correlations are changing over time. And I was wondering whether the variability depending on how we measure the variability, for example, if we were measuring sort of the variability of the signal within those same windows, whether that would be related to which metastate, quote unquote, people end up in and things like that. Because we always, whenever we have an analysis of resting state data where we're looking at dynamic functional connectivity, there's always a metastate where nobody's connected to anything, where all the networks are just sort of semi-connected, not particularly strongly. And I was wondering if that was, if we could predict that from basically the variability in the signal at any given time and whether we're really capturing the same dynamics. But there you go. But I know, Jeff, we definitely needed to move into discussions of the schedule before everyone disappears.
Yes, exactly. So, so I had this. I had this one up. I thought this would be a good one for next time. And if we tried to meet once a month, I thought this would be a topic we could discuss in late October. But after that, I think it would be great if there's sort of topics that people are interested in, especially perhaps students in the group who might want to lead a journal club, like either by themselves or in pairs. If there's something you're working on, or a recent paper, as in Stephanie's case, that just came out, directly about your research, you want to chat about it. I think this would be a great opportunity to sort of present to the group. And then also for those of you who are earlier in the program, that will have an opportunity in the spring to be able to present, at least, and doesn't have to be data, even if it's just ideas for something that you're planning on doing and you'd like to get some feedback. I think that this group could be a great opportunity for that. So are there sort of topics that jump out from today that that people would like to talk about, or papers that have come out recently that you'd like to read and discuss?
So I was wondering, because we had a good conversation, and I was wondering if we can have the same type of, at least for one or two more journal club, we can talk about the same topic. And I like the idea that we have someone to present his own work or her own work after that. I mean, this is a very nice paper. I was just mentioning that would be interesting too, if that's possible.
So are you thinking specifically in terms of variability, or sort of building upon that in different directions, or more continuing with what we've been talking about today?
I was wondering if we can probably, either way is fine. I haven't read this paper. I don't know how it is related to today's topic. It doesn't need to be. Maybe it's a good idea to have like a different topic for the journal club. That was just a suggestion.
Maybe we could circle back to the variance question in more detail in November. You know, have this one for October, since you've already read it and are interested in talking about it, but we could take give ourselves 2 months or to identify some papers and just ideas about variability.
Yeah, that sounds great.
I'm wondering, but we would need somebody to who's also interested in it, who could sort of talk through some of the other papers on it, or maybe some of the findings that might be relevant. I know what I can do. This is a good excuse for me. I need to do something on dopamine and schizophrenia anyway. So why don't I pull together some of the dopamine papers to look at what dopamine does for neural variability, and see if there's some papers there that we could talk about.
Yeah, I think that would be great.
I would be coming at it from a schizophrenia point of view, but if you give me a week to find some papers and see what's there, and then we can send it out.
Well, and that's, a lot of that is what I left out today, because I'm coming from the cognitive perspective, but there's clinical utility of variability as well. And there's been a history of studies in that too.
Right.
And I think some of which are in schizophrenia. I just am ignorant of that literature.
Well, you were coming at it from the cognitive side. Let me come at it from the disease side. And then we can try to figure out what are the other causes of neural stability or variability, et cetera. I'm wondering if there's anybody. I know there's a bunch of computational neuroscientists spread out across, CS and math and neuroscience, and I'm wondering if any of them deal explicitly with this sort of punctuated equilibrium idea, or these ideas of lack, variability in the signal, particularly, so that you don't have an exactly regular, they're rhythm keeping neural models, and then there's specifically non-rhythm keeping neural models. And I'm wondering if any of them have something to say on this. Maybe I forget who those folks are. Anyway, all right. So I'm up for November. But then I think for the spring, we should definitely have some of the students try to lead discussions. And I'm noticing Gabriel is here. Yay, Gabriel! But I will have to pull some of my other cognitive neuroscience students out of the woodwork to make sure that they're involved. Push them yelling and screaming into these presentations. Obviously, Sarah and Gabby, you guys are up next spring. And we'll see if there's Sarah. Lovely job today. Really, really nice presentation of your work. Very mature, in-depth description of it.
Thanks. And yeah, as it progresses, I can also come back to variability in the spring and talk more about anything else we found along the way.
Excellent. And thank you for volunteering to go first as well.
Yes. Extra points, extra stars in your crown. Excellent. And this is obviously open. I mean, this is open to everybody. So just like Lucina could come in, other students and other universities can certainly pop in as well, right? It's generic to whatever people are interested in.
Yeah, absolutely. Yeah, if you have friends, colleagues that you think would be interested in this, it'd be great to have them join.
Okay, cool. I'll definitely send it out to other folks who might be interested. We have the Emory, a bunch of Emory folks on the mailing list, but not all of them. Excellent. Well, I have to pop off too, so I'm going to run away as well.
Well, thank you everyone for, yeah, I think that was really great discussion, and I'm glad that we, Stephanie, you had a chance to share some of your work, and so I suppose we'll just sort of keep this paper for next time, but please email me or Brainspace Initiative if you have ideas about what you talk about and what you'd like to present on. Thank you, everyone.
Bye now.